Yes.
Great, thanks. Okay, so I'm Jake Zwart. I'm currently a postdoc at the U.S. Geological Survey in the data science branch. I'm going to be presenting on a GLEON project that is, boy, about 4 or 5 years in the making now, and hopefully will be submitted to soon for publication. And our group is maybe 19, 20 people and is part of the lake metabolism and carbon cycling working group within GLEON. And I'm going to be talking about how we're coupling these in-lake metabolism estimates and load estimates to infer how metabolism is impacted by stream loads. So a little bit of background, lake metabolism is the balance between carbon fixation or gross primary production and the biological carbon oxidation or ecosystem respiration. And this balance gives you net ecosystem productivity, which is a very good indicator of the lake trophic state. And whole lake metabolism is a measure of the fixation and oxidation of carbon from all organisms, including bacteria, phytoplankton, fishes, whatever's in the lake. And this metabolism is affected by and also affects concentrations of nutrients and carbon within the lake. In this simple diagram, you can see dissolved organic carbon gets respired and turned into dissolved inorganic carbon or carbon dioxide. Dissolved organic carbon also shades light and attenuates light, and so that can impact phytoplankton growth, which is grown by gross primary production, which uptakes phosphorus, and there's sediment respiration. So there's a lot going on that in metabolism is essential to understand dynamics of lake systems. Metabolism is also an indicator of the lake state. It impacts food web structure, energy mobilization through these different trophic levels, rates of carbon accumulation within the sediments, and carbon dioxide and methane emissions from the lake surface, and how lakes fit into the global carbon budget. It's essential to understand variations in lake metabolism and what's controlling lake metabolism. So as I mentioned, concentrations of carbon and nutrients impact lake metabolism. And this has been known for a long time, and a lot of in-lake variables such as carbon and nutrients have been used to predict lake metabolism, including a few GLEON projects. This is just an example of total phosphorus being a pretty good predictor of gross primary production. Phosphorus is often a limiting nutrient of algal productivity within lakes, and you can see this in the Solomon et al. Data set that looked at across 25 lakes within the GLEON network. Another example from Hansen et al. 2003, where again total phosphorus is a pretty good predictor of gross primary production. Also, dissolved organic carbon is a pretty good predictor, positively correlated to ecosystem respiration. So it's great that these in-lake predictors are pretty good predictors of lake metabolism, but there's still a lot of variation that's left unexplained. And we also know that these in-lake nutrient concentrations are impacted by the surrounding catchment. And so we're trying to step outside of the lake and into the catchment and include some of these catchment predictors into estimates of lake metabolism. So we know that nutrients and carbon come from the surrounding landscape. It's also important to include these in our predictions of lake metabolism because they can respond a lot to changes in climate and land use. And so understanding how these loads impact metabolism is important for projecting lake metabolic state into the future under climate and land use changes. So climate change not only impacts lake properties such as water temperature and stratification strength, for example, but these catchment properties are also influenced by climate change, such as terrestrial growing, season length, stream discharge, soil properties, etc. And land use changes influences stream loads, mainly through its effect on stream nutrient and carbon concentrations. For example, like if everything is converted into agricultural lands, there would be an increase in nutrient concentration within the stream, which would get transported to the lakes and likely impacting lake metabolism. So we're trying to make understand the variation between stream loads and metabolism to better make projections of how metabolism may change under these various climate and land use changes. And there's a growing body of work that's stepping out of the lake and into the catchment and making predictions of how loads impact lake metabolism. This is an example from a theoretical paper from Kelly et al. In 2018. They looked at concurrent effects of dissolved organic carbon and nutrient loads and its impact on pelagic primary production. The model predicted that with increasing dissolved organic carbon load, there's initially an increase in primary production, or GPP, followed by a decline when DOC is very high. This is because there's the nutrients that are associated with this dissolved organic carbon load, and as you increase the DOC load, you simultaneously release algae from nutrient limitation while increasing light limitation. And so then they predicted that the peak and the magnitude of this peak is dependent on the load stoichiometry and the chromophoricity. And so this is all just a theoretical model and hasn't been tested with real data until Bergstrom and Carlson looked at 25 lakes or so and show that, yes, this lake survey, cross lake survey, did reproduce some of these patterns, these hump shape patterns, and showing that the stoichiometry of the load really impacted the magnitude of where this peak happened. And so this figure is just showing that you get these hump shape patterns, and this influence of the stoichiometry of the load is impacted by this nitrogen enrichment and nitrogen and phosphorus enrichment, where nitrogen enrichment increases the magnitude of the, at least in this case, the phytoplankton biomass, and NNP enrichment increases it even more. So this is great, but we would really ideally have a data set that couples stream loads and metabolism rather than these artificial nutrient enrichments, and then we can test some of these predictions of how loads impact lake metabolism. And furthermore, if we had a data set with inflowing loads and in-lake nutrient concentrations and metabolism, it would not only allow us to examine the effect of metabolism on the effect of load on metabolism, but the effect of metabolism on freshwater stoichiometry, for example, carbon to nitrogen, carbon to phosphorus, nitrogen to phosphorus, and how metabolism affects this landscape scale carbon nutrient cycling along this aquatic continuum. So our big GLEON group has done a lot of work trying to assemble the 16 lake data sets, lake and stream data set, where we're coupling lake metabolism and stream load estimates. This is a map of all the lake locations. You can't really see all 16 dots just because some of the lakes are clustered close together. But about half the lakes are in North America and half the lakes are in Europe. We're a little bit biased in the Northern Hemisphere. All lakes occur above 35 degrees north. A little bit of a description of our data set. Again, we have daily estimates of stream carbon, nitrogen, and phosphorus loads, along with lake metabolism, so gross primary production, respiration, and net ecosystem production. The lakes vary in their lake and catchment characteristics. The number of inflows varied from no inflows to 6 inflows. Lakes, we had 2 lakes without inflows, and we couldn't estimate loads for those lakes. Maximum depth varied from 5 to 45 meters, surface area 0.8 to 27,000 hectares, water residence time from very short 0.1 years to 5.3 years, and catchment properties varied from forested to agriculture to wetland, although most of our catchments were pretty heavily forested. Our load estimates, we're estimating stream load for carbon, nitrogen, and phosphorus. Again, these were estimated by multiplying daily water discharge by daily concentrations of nitrogen, phosphorus and carbon. And these concentrations of carbon, nitrogen, and phosphorus were taken from daily to monthly grab samples, and then these nutrient concentrations were linearly interpolated to daily scale. We didn't do any flow-weighted concentrations because that didn't improve any of the, there was no relationship for with flow for any of the stream inflows. For our lake metabolism estimates, we're estimating metabolism using the R package lake metabolizer and fitting models to diagonal curves of oxygen. Background on lake metabolism, oxygen tells us about the rates of photosynthesis and respiration because photosynthesis produces oxygen and respiration consumes it. And we also estimated uncertainty in our metabolism rates using a bootstrapping routine, and we use these bootstrapping estimates and these uncertainty estimates to filter out days that didn't fit well. So with this data set, we're asking a few questions. First of all, like, what is the variation in metabolism loads across these lakes? There hasn't been a really good data set with inflowing concentrations, loads, and stoichiometry coupled with in-lake stoichiometry. And so we're just comparing the load and in-lake stoichiometry and seeing if that changes or not. And comparing our load stoichiometry to previous survey values to put it into context. And then finally, the question that we're really interested in is like what load metrics best predict metabolism. For this analysis right now, we're just aggregating our daily estimates to an annual mean and then using a KICS information criteria, AIC, to pick out a best model using all of our load metrics, including carbon load, nitrogen load, phosphorus load, and then the stoichiometry metrics, C to N, C to P, and N to P, to predict gross primary production, respiration, and net ecosystem productivity. Okay, so I'm just going to jump into more of the results and the dataset description. This is going to be looking at kind of the load variation across lakes and within lakes through time. I'm just going to show you one example of DOC load for all the lakes. You can see that there's 14 of the 16 lakes that had dissolved organic carbon load estimates. Crampton Lake and Nashtarn did not. And let's see, this is across a single year for each lake. We've colored the lines of the DOC load based on the season, so spring, summer, and fall. And you can see there's a lot of variability across the lakes and within the lakes through time, which these pulses of dissolved organic carbon load may impact the lake metabolism estimates. It's also important to note that these DOC loads are weighted or volumetrically weighted to the lake epilimnion, and the lakes are plotted in increasing average gross primary production from left to right to top to bottom. So Trout Lake in the top left is the lowest GPP estimates, and Acton Lake in the bottom right is the highest GPP estimates in our data set. I'm not going to show you N to P plots, or N and P loads, because they're pretty similar to this. Again, we're using aggregating most of these daily estimates to annual scales, and just give you a flavor of how these loads correlate with each other. DOC and total nitrogen and DOC and total phosphorus loads did not correlate with each other. There's quite a bit of variation within our data set. So each point on these graphs is a lake. And then, maybe not surprisingly, total nitrogen and total phosphorus correlated pretty strongly with it, positively correlated. The graph has a lot of information, but I'm going to try to unpack it. And so it's looking at how both the in-lake stoichiometry and load stoichiometry vary across lakes and within lakes through time. So the load stoichiometry, so C to P, which is the black lines or points, N to P, which is the pink points, and C to N, which is the orange points and lines. The loads are given by the lines. The in-lake stoichiometry is given by the points. And then there's these dotted lines across each of the panels, and those are kind of a reference line for Redfield ratios. So C to P at 106 and N to P at 16 and C to N at 6.6. So you can see most of the lakes, especially the ones with lower GPP, are pretty high above these Redfield ratio lines, especially for carbon to phosphorus. And then Lake Mendota, Acton Lake in the bottom right panels are getting a little bit closer to those Redfield ratios of 106. And even below the Redfield ratios for carbon and nitrogen. So this is a really busy graph. We can take a look at a couple of lakes just to give you a little bit more detail. You can see that again, the load stoichiometry is the lines, the in-lake stoichiometry is the points. And for the most part, the lake stoichiometry matches the load stoichiometry fairly well. And so Acton Lake has the highest GPP, Harp Lake has a pretty low GPP, Acton Lake is a very low residence time, Harp Lake is a longer residence time. You can see that as load stoichiometry changes in Acton Lake through time, the in-lake stoichiometry also changes. It's most prominent for carbon and nitrogen, the orange line, and nitrogen phosphorus, the pink line. And again, the stoichiometry is much lower than compared to the Harp Lake, which is much higher. But again, for right now, we're aggregating a lot of these daily estimates into annual scale. And we can look across lakes and see how the aggregated carbon to phosphorus, carbon to nitrogen, nitrogen to phosphorus stoichiometry compares to the lake stoichiometry. And for our lake data set, we have a very high C to P ratio for load and lake. Load stoichiometry, median C to P is 3,000, a little over 3,000. Median lake is a little over 2,000. And most the load stoichiometry is lower than the lake stoichiometry. And we compare this to a paper that was published in 2018. They looked at median US streams and median US lakes for C to P. We're much higher above those C to P estimates. And it's also good to note that this previous survey, they saw an increase in C to P going from streams to lakes, where we see a decrease from C to P going from the streams to the lakes. Pretty similar story for carbon and nitrogen. Again, our carbon and nitrogen stoichiometry for both the load and the lake is pretty high. And we see a decrease in C to N from the stream to the lake. Again, this is in contrast to this US survey where the stream C to N increased from when going into the lakes. Nitrogen and phosphorus was kind of all over the place, but median load was again higher than compared to this US survey. We had 84 of nitrogen and phosphorus and 72 of nitrogen and phosphorus for the lake. Again, decreasing from going from the stream to the lake, but there's a lot of variability in the nitrogen and phosphorus load stoichiometry compared to the lake stoichiometry. So now we're going to dive into some of this metabolism results. The metabolism varied a lot across all the lakes. Again, Trout Lake being the lowest metabolic rates and Acton Lake having the highest. Again, this is colored by season, so spring, summer, and fall. And then you can see that generally the highest GPP occurred during the summer seasons. And to orient you a little bit more to this, gross primary production is positive, so above those dotted lines, and ecosystem respiration is negative, so below those bottom that dotted line. Again, we're aggregating all these daily estimates into an annual mean, and then asking which load metrics, so the carbon, nitrogen, phosphorus loads, and also the stoichiometry of the loads, which model best predicts metabolism. So diving right into that, best predictor for GPP across the lakes is total nitrogen load and dissolved organic carbon load. Predicts GPP pretty well at the annual scale, at least, with R squared of 0.8. We don't have another data set to validate, and so we're just calibrating on the data set that we have. But the coefficient on total nitrogen load was positive, coefficient on dissolved organic carbon load was negative, showing that the positive impact on nitrogen and negative impact on DOC on GPP. Total nitrogen load and dissolved organic carbon load was also the best model for predicting respiration. This, again, with pretty high R squared of 0.84. This is maybe not too surprising to have a similar model because GPP and R are very highly correlated across these lakes. And we didn't do very well for predicting net ecosystem production, R squared of 0.15, and the load nitrogen to phosphorus stoichiometry was the best model for predicting net ecosystem productivity, but it was only slightly better than just the intercept model, so we're not putting too much faith into this. So that's a little bit of a flavor of what our groups have been up to. Again, we've been aggregating everything to the annual scale, and some of our next steps, which would probably be a different paper, would be to look at some of this temporal variation in load and metabolism. How does the load timing, like, you know, does a big spring pulse or a summer pulse, does it impact metabolism differently? And then also, how does metabolism and water residence time impact stoichiometry and the cycling of nutrients across this water continuum? So I think I'm a little bit over time, but that's end of my presentation. I'll take any questions if there are any.
Thank you very much, Jake. Very interesting work. Do we have quick questions for Jake? If not, maybe I can ask one again. It didn't really come up, but the thought just, you know, popped into my head. If you're interested in long-term time series of lake metabolism, is that something that is at all possible to get an idea of that, or is that methodologically too difficult?
I mean, there are some lake data sets that have a long metabolism time series, but trying to think.
Don't you suffer then from changes in methodology over the years?
Yeah, that's another thing. So we're using the free water method, which I'm guessing the longest data set for that is maybe 20 years, but that's maybe 1 or 2 lakes. And then there's other methods. There's been papers that have compared the different methods like bottle incubations and like C13 method to the free water, and you can get different estimates, and so it's not exactly apples to oranges all the time, but you could string together some of these estimates to get a long time series.
Okay, cool. Thanks again, Jake, and thanks Laura also for 2 presentations. For me, this is, you know, a good example of what one of the aims we have with the 0.5 meetings, that we actually now had the opportunity to listen to the outcome of 2 projects that many of us, you know, are aware have been ongoing for quite a few years, and now, you know, we get to listen to the outcome of them. I think